Good morning. I am Gisele Monteiro from the University of São Paulo, and I will talk about the characterization and protein engineering of L-asparaginase 1 from Saccharomyces cerevisiae to evaluate its use as a potential biopharmaceutical. First, I will introduce you briefly, because we are in a cancer summit, from leukemia. Leukemia is a blood cancer that prevalence in childhood is 80% of cases, and the treatment includes chemotherapy and the enzyme L-asparaginase, that is the biopharmaceutical. And in most cases, at least in Brazil, we have complete remission and cure using adequately this chemotherapy, including the biopharmaceutical. The discovery of this biopharmaceutical, the L-asparaginase, was performed by Kidd in 1953 using serum guinea pig to cultivate leukemia cells. This researcher observed that this serum possessed something that kills cancer cells and not the normal blood cells. Lately, several other researchers followed, and the isolated factor that is responsible for this anti-tumor effect of serum from guinea pig was characterized as the L-asparaginase. And what this enzyme does? The L-asparaginase is an hydrolase enzyme, and it is responsible for cleave, hydrolyze this amino group, and generate L-aspartic acid and ammonia. And in a less extent, is also responsible for cleave the L-glutamine amino acid, corresponding to this asparaginase action and this glutaminase action. In a less extension, but producing L-glutamic acid and also ammonia. And what this is, why this is related with cancer? The anti-tumor activity of this enzyme is due to the fact that several types of tumor, but especially in leukemia, we have this scenario that cells, leukemia cells and not normal cells, possess epigenetic silence in the asparaginase synthetase gene. So this enzyme is not expressed, and the cancer cells are not able to produce endogenous asparagine, being strictly dependent on bloodstream asparagine from the diet. So when you deplete the serum asparagine using injection of this enzyme in the blood, the cancer cell suffers starve and have problems with translation of proteins, including anti-apoptotic proteins and proliferation proteins, and becomes in a process of apoptosis. This scenario is also true when we talk about the cells that possess low levels of asparagine synthetase expression of this enzyme. These cells are also sensitive to L-asparaginase treatment, but in some cases that they are resistant to L-asparaginase treatment are some patients that present leukemia cells that express high levels of asparagine synthetase enzyme. So in that case, the asparagine is not effective as treatment. So the first characterized biopharmaceutical was in guinea pig, but it was really difficult to obtain high amounts, industrially cost-effective from that source, because when we tried to purify the enzyme from the serum, was too much contamination, and the enzyme was inactive in some case. In 1964, other researchers characterized the bacterial isoform that is able to produce through fermentation and isolate easily the enzyme from the bacteria. And to be a biopharmaceutical, these enzymes need to show some characteristics, mainly kinetic properties, such as low Michaelis-Menten constant, because asparagine blood stream concentration is around 50 micromolar, so the enzyme needs to be in micromolar range to be effective, to deplete the asparagine. And the specific activity, that is the in international units, micromole by minute by milligram of protein of asparagine catalyzed, hydrolyzed, needs to be around this number, 200, and between 100 and 200. Actually, we have three main formulation of this enzyme. Two from Escherichia coli, isolated from this. The native one, one of the marks trade is L-aspar, and the pegylated isoform of Escherichia coli enzyme, the oncaspar. And the other is the erinase that is isolated from the other species of bacteria, Erwinia chrysanthemum. And this specific activity corresponds to the dose necessary to be effective in deplete asparagine from the bloodstream. So you can see here that with a lower specific activity, you need more units to have the same anti-tumor effect. Problems with bacteria, and as I starting to say, the treatment with asparaginase is really effective. 90% of the cases is suffer complete cure and remission. But there are some problems in use bacterial enzymes in human. The first one, easy to imagine, is the allergic reactions. Bacterial enzyme asparaginase is a tetramer of around 120 kilodaltons. So it's an enormous enzyme applied, directly injected in human bloodstream. And so following, this is an arm for a child, following the weeks we can see some strong allergic reactions. And these allergic reactions is one of the motives that in adult is not recommended to apply asparaginase, because the immune system is high activated, and adults can be more prone to suffer anaphylactic shock. Hyperammonemy. As I showed you, the enzyme catalyzes the hydrolyzation of the amino group, and this amino group, the ammonia, is leave. So hyperammonemy is a very toxic side effect of the treatment with this enzyme, causing sometimes growth retardation, several toxic to nervous system, and very toxic effects to liver. And the other are immune reactions caused by the formation of antibodies and proteins. What is this? When you treat a human with this enzyme during the time of treatment, immune system creates antibodies specific to this enzyme, the asparaginase. And this usually opsonize the enzyme and inactivate it. In addition, bloodstream protease, specifically asparagine endopeptidase and cathepsin B, are responsible for cleave this enzyme in the bloodstream and causing a silent inactivation. So the patients do not suffer symptoms of hyperammonemia or allergic reactions, but they are not effectively being treated because of silent inactivation of the enzyme. These are just to represent some titles of titers of antibody in levels causing in patients with allergy and or silent inactivation, high levels of antibodies, or without silence or allergy. And here showing that when you add PEG to the enzyme, polyethylene glycol, we have lower allergic reactions. So the objective of this work was to test the potential of asparaginase 1 of Saccharomyces cerevisiae as a biopharmaceutical to treat acute lymphoblastic leukemia. We have just little studies about this enzyme, accounting for 1970, and they just characterized the endogenous enzyme from Saccharomyces cerevisiae. And the purification steps were several and hard, so showed at the final very little specific activity and not good kinetic parameters. But because of this methodology, we asked this is true, so we tested again in a recombinant expression system. Just to show you that is promising, we have analyzed the amino acids that are responsible for catalytic, that are part of catalytic cycle in the bacterial isoforms used in the treatment today. So you can see that our enzyme has the complete conserved active site amino acids. So we cloned the gene in a bacterial expression system. Among these restriction enzyme sites, and overexpressed this enzyme in fusion, in any terminal fusion with a his-tag, what allows us to purify the enzyme in low steps of purification. Just to be true, just one step, that was immobilization of metal IMEC. And these are the new parameters to the purified enzyme. We obtained a specific activity of up to 200 international units by milligram of protein. The enzyme was not a Michaelian one because the parameters was plotted very well in Hill plot, so these show behavior of an allosteric enzyme that is interesting. After I will try to convince you about this. The pH, the optimum pH is and the temperature optimum for these enzymes are according to the use of humans, 7.5 and 37 degree. And here are, as the enzyme is not a Michaelian enzyme, we cannot call Michaelian constant, so we have a K prime that is around 75 micromolar. That is around, that is split good. And the specific activity and the good catalytic efficient with an allosteric coefficient with positive of 2. This means that this enzyme is regulated by the concentration of the substrate. And one of the most important things that I consider that this enzyme has no glutaminase activity. And why this is important? This second activity of L-asparaginase leads to production of more ammonia, because L-glutamine is one of the most abundant amino acids present in the blood serum. So when you have an enzyme that does not possess this activity, less chance to reach hyperammonemia. Here was just to characterize that amino acids that participate in the active site. Mutation of them, all of them, caused more than 99% of loss of activity, showing that all of them are related to the active site. And these mutations were not mutations were not responsible to unfold of protein. They are related to loss of ability to perform catalysis, because the circular dichroism show that fold of protein are equal to the non-mutated one. And the most important thing, this enzyme has the parameters, kinetic parameters adequate, the activity specific, but it has an anti-tumor activity against leukemia. So we tested in vitro to say that this is promising. We use in cytotoxicity assay with multi-4 leukemia cells, and we see that yes, we have an anti-tumor drug, that the biopharmaceutical, because this was reduced the leukemia viability less than around 20%. Just to compare, we bought Kidrolas, that is formulated asparaginase, is clinical used asparaginase, and the levels were the comparable. Of course, here is a formulated drug, so it's better. The cure to less than 10% of survival in leukemia cells. But we think that when we optimize the conditions of purification, stabilization, and activity of this enzyme, we could reach this level. So the concluding remarks are, the enzyme from Saccharomyces cerevisiae has potential to be a new biopharmaceutical to treat acute lymphoblastic leukemia, because the enzyme possesses high specific activity, no glutaminase activity. It's an eukaryotic protein, so it's, for a while, is just a speculation, but we hope that this enzyme presents less epitopes to immune system. The fact that it's an allosteric enzyme, this is a good useful to control the enzyme, because when the enzyme reaches the maximum velocity, is needed the high concentration of the substrate. So when the substrate concentration low in the bloodstream, the enzyme stops. So this allows less hyperammonemia. We are testing this also experimentally. And the high substrate affinity, because the bloodstream concentration of asparaginase. And the most important, we have proved that this enzyme possesses anti-tumor activity. So these are the authors of this work. I say thanks to all the authors and the 3 last are professors. This is our coordinator of the thematic process, financed by FAPESP, our foundation, Adalberto Pessoa Jr., and this is my research group, and thank you all for attention.